Okay, so um, we talk a lot about oxygen being transported in blood via hemoglobin and the structure of hemoglobin and the binding of oxygen and the oxygen dissociation curve and things like that. But and so it's easy for students to forget that carbon dioxide is also transported in the blood, and the transport of this is just as critical as the transport of oxygen because it's a waste product. So it needs to be taken from the respiring tissue to the lungs, where it then diffuses out of the blood and into the alveoli to be breathed out. Okay, and so when we're talking about carbon dioxide transport in the blood, there are 3 ways that carbon dioxide can be transported in the blood. The first way is that it can be dissolved in the plasma. In the liquid part itself of the blood. Okay, so it can be dissolved in the plasma. The second way is bound to hemoglobin. So that is, let's see, give you the name of it. So it binds to hemoglobin. So this is once the hemoglobin has dropped off its oxygen, then it is capable of binding to carbon dioxide, and that is called to form carbaminohemoglobin. Sorry. Okay. And then the third way is that it is transported in the cytoplasm of the red blood cells themselves. So not in the hemoglobin, but in the cytoplasm of the red blood cells. And it's transported as hydrogen carbonate ions. And I'll walk you through how that happens. All right. So in the cytoplasm of the red blood cells, and I'll spell out hydrogen carbonate ions once, but then I won't be doing it again. Okay, and just to give you an idea of how much, this is not evenly shared, right? It's not like a third of each. Okay, so only about 5% is carried in the plasma, right? And then another 10 to 20% is going to be found in the in the form of carbaminohemoglobin. So this is going to be, I don't know, 10 to 20%. And the remainder, so we're talking between 75 and 85% is carried as the hydrogen carbonate ions in the red blood cells. Okay, so then let's just briefly look at how does this actually happen. Oh, can you see yeah, you can see that. Okay. So the idea is that the carbon dioxide is when it dissolves in the water. Okay, so when you have carbon dioxide and water molecules, these can combine, and I'll be careful about how I draw this, to form something called carbonic acid. All right, so obviously it's a combination, so this is its chemical formula, and this is called carbonic acid. Okay, and I put this because it's important to understand that this is a reversible reaction. It's not like the CO2 when it combines with the water is then like in this form of acid that is it is permanent. Okay, so the idea is that this reaction is reversible. Okay, those of you in chemistry would understand that symbol. And then once you have the carbonic acid, because it's still surrounded by water, it will dissociate. Okay, and it will form the hydrogen carbonate ions. Okay, as well as hydrogen ion. Okay, and so this is also a reversible process. Okay, but the key thing is, so this process cannot be controlled. Once you have carbonic acid and it's surrounded, it's in an aqueous environment, it's naturally going to dissociate into the hydrogen carbonate, which I will label. So these hydrogen carbonate ions, that's what this is. Okay, and so this is going to naturally form. So the key thing to control here is this reaction. And this is controlled by an enzyme called carbonic anhydrase. Okay, and so And this enzyme, when I say it controls it in both directions. So it can either increase the amount of carbonic acid, or it can take the carbonic acid and turn it back into CO2 and water. All right. So this enzyme is the one that controls this. And so then, thus, by controlling the rate of this reaction and how much carbonic acid around, then you can indirectly control how many hydrogen carbonate ions you have around. Okay. So the idea is that when you're in respiring tissue, for example, all right, and you have a large amount of CO2 around, then this enzyme is going to take that carbon dioxide and convert it, with the help of water, into the carbonic acid. And just to be clear, this whole process, right, the enzyme itself is found in the cytoplasm of the red blood cell. So this is happening in the cytoplasm. All right, the idea is that the CO2 diffuses into the red blood cells. Oops, sorry. And then this enzyme is in the cytoplasm of the red blood cells, and then catalyzes this conversion. Or if you're looking at what's happening in the lungs, it's catalyzing the carbonic acid to move back to CO2 and water. And so the idea is that what you end up with then are these hydrogen carbonate ions. And remember, that's how the vast majority of carbon dioxide is carried in the blood, plus these hydrogen ions. Now, oops, press the wrong button, sorry guys. So these hydrogen carbonate ions then diffuse out of the red blood cell. Okay, and it's a diffusion, so it's just the concentration gradient. This is not an active thing. Okay, and these are ions, so if you had to think about it, you should probably understand that perhaps there are channel proteins that are allowing this diffusion to happen. Okay, so this would be facilitated diffusion. And so these ions diffuse out of the red blood cells, but the problem is that that leaves a whole bunch of hydrogen ions inside, as well as introduces a large, if a lot of these are diffusing out, then you can start to create a shift in the pH of the blood. And so in order to combat that, if you have negatively charged ions moving out of the cell, you need to have some sort of negatively charged ion moving into the cell. And so in order to combat that, that's exactly what happens. And so the idea is that you have chloride, which is also a negatively charged ion. So chloride ions are going to move into the cell in order to maintain the proper balance of charges and pH. And this is called, the idea of these chloride ions moving in, this is called the chloride shift. Okay. And so they're moving into the cell from the plasma. Okay. The other thing is, so that's number 3, and that's how the majority of how it's covered. Now, I said that 10 to 20% binds to hemoglobin to form the carbene-aminohemoglobin. And so the idea with that is that happens, I should have drawn this better. That's what happens over here. So just like you should be worried about this diffusing out of the red blood cells and causing a change in pH, these can also lead to a change in pH. And so what happens is the fate of these, I apologize, I'm going to draw this over here. The idea is that these ions, they combine with hemoglobin, which I'm going to abbreviate HB, which I've done before in class. So they combine with hemoglobin, and that forms hemoglobinic acid. And basically what this does is that it acts as a buffer. Because again, you do not want the pH to be changing either within the cells or out. pH is critical for so many different processes, and so there's a lot of mechanisms in your body and in certain cells to prevent too much change in pH. So that's what this is about. Okay, both the chloride shift and the formation of the hemoglobinic acid. Right, so that's how CO2 is transported in the blood. Sorry, I realized that was underneath the cords. And like I said, we haven't seen many questions on this, so it's not something that I can direct you for in terms of the mark scheme. But the other thing to keep in mind is that how this works in relation to the Bohr effect. So remember that the Bohr effect is basically how does increasing levels of CO2, the presence of CO2, affect the affinity of hemoglobin for oxygen? Okay, so I'll write that down. I'll pause it while I write that down. Okay, so the Bohr effect, remember, it's the change in the oxygen dissociation curve, and it's not just any change. It's the changes that result because there are different levels of CO2 around. And in general, if I were to draw a very rough oxygen dissociation curve, where I have the partial pressure of oxygen in kPa, and then this is the percent saturation. The idea is that if you have your regular curve here, sorry, it should be more sigmoidal, but it's not. Let's pretend that is, okay? As you increase the amount of CO2 that is present, the partial pressure of CO2, you're going to get, the curve is going to move down, okay? And so, I should eventually come back up here. And so the idea is that as you increase, you're going to get more and more looking like this. And remember that when an oxygen dissociation curve moves down and to the right, it is decreasing the affinity, okay? And so the idea is that as you increase the amount of CO2, you are decreasing the affinity of hemoglobin for oxygen. And the reason for that should make sense if you look back at this, because even though your cells are doing the best they can to control the pH will change slightly because you have these acids around, okay? And so that will lower the pH, and then hemoglobin, because it's a protein, it's sensitive to changes in pH, and therefore its affinity for its target, which is O2, percent saturation of hemoglobin with O2. So the affinity will decrease, all right? So you should understand, so because this has come up, the Bohr effect has come up, and I think it's important to understand how carbon dioxide is transported in the blood in order to kind of make sense of the Bohr effect. If people want me to do a video on the Bohr effect, let me know, and I will add that to my list.